बायोटेक्नोलॉजी से है, जिसका कि एंजाइम का एमसीक्यू देखेंगे, एंजाइम बायोटेक्नोलॉजी एंड बायोसेंसर का। ओके, ये 6 सेम के लिए है और इम्पोर्टेंट इसलिए है क्योंकि जो एग्जाम हो रहे हैं ऑनलाइन एग्जाम, उसमें बहुत ज्यादा इम्पोर्टेंट है। ओके, तो चलिए देखते हैं फर्स्ट क्वेश्चन नंबर। Which of the following scientist was discovered mold penicillin? कौन से साइंटिस्ट था जो मोल्ड पेनिसिलम को डिस्कवर किया था? तो इसका जो आंसर होगा ए होगा अलेक्जेंडर फ्लेमिंग। ओके, लुईस पास्चर, इसका कुछ डिस्क्रिप्शन देख लेते हैं। लुईस पास्चर जो है डिस्कवर किया था प्रिंसिपल ऑफ वैक्सीनेशन, माइक्रोबियल फर्मेंटेशन एंड पैस्चराइजेशन। ओके, एम गिलर्ड जो है वो आइडेंटिफाइंग ऑफ डीएनए ग्लेज वो 1967 में हुआ था। एंड देन कैम वेजिमन जो है वो मेड प्योर माइक्रोबियल माइक्रोलॉजिकल प्रोसेस इन एन इंडस्ट्रियल प्रोसीजर 1917 में किया था। नेक्स्ट सेकंड क्वेश्चन के नंबर की बात करेंगे। Which of the following has all virus consist of? The answer is D, nucleic acid and protein coat. The answer is D. Okay, next is third question number. Application biotechnology procedure in medical process is classified as? The answer is B, red biotechnology. इसका कुछ एक्सप्लेनेशन देखते हैं रेड का, व्हाइट का। इसका व्हाइट का बायोटेक्नोलॉजी का क्या होता है? ऑल्सो नोन एज इंडस्ट्रियल बायोटेक्नोलॉजी। ब्लू बायोटेक्नोलॉजी का बेस्ड ऑन द एक्सप्लोरेशन ऑफ सी सोर्स टू क्रिएट प्रोडक्ट्स एंड इंडस्ट्रियल एप्लीकेशन। एंड देन ग्रीन बायोटेक्नोलॉजी की अगर देखें तो बायोटेक्नोलॉजी अप्लाइड टू एग्रीकल्चर प्रोसेस में ये काम आता है। ओके, इसका आंसर जो है वो बी होगा। नेक्स्ट जो है फोर्थ क्वेश्चन नंबर, विच ऑफ द फॉलोइंग इज एन एग्जांपल ऑफ येलो बायोटेक्नोलॉजी? ओके, इसमें कौन सा एग्जांपल है येलो बायोटेक्नोलॉजी का? तो इसका जो आंसर होगा ये होगा यूज ऑफ द बायोटेक्नोलॉजी इन फूड प्रोडक्शन। ओके, अच्छा इसमें अब ये देखते हैं कि ग्रे बायोटेक्नोलॉजी क्या होगा? डेडिकेटेड टू इन्वायरमेंटल एप्लीकेशन। देन वायलेट बायोटेक्नोलॉजी क्या करता है? लॉ, इथिकल एंड फिजियोलॉजिकल इशू एंड अराउंड बायोटेक्नोलॉजी। ओके, इसका जो आंसर होगा ए होगा। नेक्स्ट देखते हैं 5 क्वेश्चन नंबर। द यूनिट प्रोडक्ट ऑफ ट्रांसलेशन इज पॉलीपेप्टाइड्स। द आंसर इज सी, पॉलीपेप्टाइड्स। ओके, इसका कुछ एक्सप्लेनेशन है। द अमीनो एसिड सीक्वेंस इज द फाइनल रिजल्ट ऑफ ट्रांसलेशन एंड अल इज अ polypeptide. Polypeptide can then undergo folding to become functional proteins. Okay, the answer is C. Next is 6th question number. Restriction enzyme were discovered by Smith and Nanthans. Smith and Nanthans. The answer is A. एस्क्लेंडर फ्लेमिंग के बारे में बात करें, डिस्कवर्ड किया गया था मोल्ड पेनिसिलम। एंड बर्ग, बॉयलर और कोलिन जो है वो डिस्कवर्ड किया था डीएनए क्लोनिंग टेक्निक्स को। ओके, नेक्स्ट बात करते हैं 7 क्वेश्चन नंबर। यूएस अप्रूवल फॉर फर्स्ट फार्मास्यूटिकल प्रोडक्ट डिराइव्ड फ्रॉम डीएनए टेक्नोलॉजी वाज ह्यूमन इंसुलिन। द आंसर इज बी, ह्यूमन इंसुलिन। ये क्या हुआ? ह्यूमन इंसुलिन जो है ह्यूमलिन भी बोल सकते हैं, इन जेनेटिक एंड इलीरी एंड सीओ 1982 में हुआ था। ओके, नेक्स्ट इज 8 क्वेश्चन नंबर। द ह्यूमन जीनोम इस प्रोजेक्ट वाज लॉन्च इन द ईयर। कौन सी ईयर में हुआ था? 1990 में। ओके, इसके बारे में कुछ जान लेते हैं। ह्यूमन जीनोम प्रोजेक्ट जो है इसका शॉर्ट फॉर्म एचजीपी होता है। वाज अ इंटरनेशनल साइंटिफिक रिसर्च प्रोजेक्ट which got successfully completed in the year 2003 में हुआ था। ओके, कंप्लीट हो गया था। नेक्स्ट बात करते हैं 9th क्वेश्चन नंबर। द restriction of enzyme mobility in a fixed space is known as? The answer is A, enzyme immobilization. enzyme immobilization क्या होता है? The restriction of enzyme mobility in a fixed space is known as enzyme mobilization. Immobilization. Immobilization does not always means immobile. It means confining an enzyme to a confined space. Okay, next is 10 question number. Which of the following is not a property of carrier matrix? कौन सा है नहीं, कौन सा नहीं है कैरियर प्रॉपर्टी मैट्रिक्स का। तो इसका जो आंसर होगा थर्मल matrix should have the following properties: stability, physical strength, and easily available. Thermal stability of an enzyme increases is an advantage of immobilized enzyme over free enzymes. Okay. Next is 11 question number, in which the following method the enzyme is bound to a suitable absorbent material making it immobile. The answer is B, adsorption. इसका कुछ ये देख लेते हैं. In covalent binding method, the enzyme is immobilized to matrix by covalent interaction between them. Then second is in entrapment method देखते हैं. The enzyme is entrapment method with a suitable gel or fiber. And then अब बात करते हैं membrane confinement का. Enzyme in aqueous solution is confined with a semi-permeable membrane. ओके इसका जो आंसर होगा वो बी होगा. ओके नेक्स्ट की अगर हम बात करें 12 का, which was the first enzyme to be immobilized by adsorption method. इसका जो आंसर होगा सी होगा, इन्वर्टेज. इसका कुछ एक्सप्लेनेशन है वो देख लेते हैं. The first enzyme to be immobilized was invertage. It was immobilized on activated charcoal, but it was not used for the further any process. This is what done by Nelson and Griffin in 1916. Okay. Next, 13 question number. Which of the following matrix is not used while immobilizing the enzyme by covalent binding? The answer is C, calcium agglutinate. Okay. इसका कुछ explanation है. The most commonly used matrix are agarose, cellulose, polyacrylamide, and glutaraldehyde and trialkoxy silanes etc. Calcium agglutinate is used for the entrapment method of immobilization. Okay. Next is 14 question number. Which of the following is not a matrix used for the entrapment? The answer is C, glutaraldehyde. The answer is C. इसका कुछ explanation है वो देख लेते हैं. Entrapment is a method which involves the entrapment of the enzyme within the suitable gel or fiber. Entrapment can be achieved in two ways: polymerized gel entrapment and microencapsulation. The matrix is used entrapment are agar, collision, and diatomaceous earth and activated carbons, porous ceramic calcium aggregate, and K-carnitine etc. Glutaraldehyde is the matrix is used the immobilization by the covalent binding. Okay. Next is 15 question number. Entrapment of calcium agglutinate is not used in case of which of the following? The answer is C. कौन सा होगी? C. Immobilization of the invertase enzyme. Okay. Immobilization of inverted enzyme is done by using activated charcoal adsorbent. This is adsorption method is immobilization. The answer is C. Next is 16 question number. Which of the following method can be applied when substrate molecules are much smaller than enzyme? Hollow fiber. The answer is C, hollow fiber. Okay. Kuch explanation of hollow fiber is the method which can be applied when substrate molecules are much smaller than enzyme. Hollow fiber is method used in membrane confinement. The answer is C. Okay. Next is 17 question number. Which of the following is a disadvantage an immobilized enzyme? The answer is B. Immobilization means additional cost. The answer is B. इसका कुछ explanation है. The disadvantage is an immobilized enzyme is additional cost. The first advantage, immobilization allow continuous process. B is productivity increase as it may used fully at higher concentration and for a longer time. Then third is immobilization prevent loss of activity as immobilized enzyme cannot be denatured. Okay. Next is 18 question number. The effectiveness factor increases with. The answer is A. Increase of diffusivity and decreased with the increase of particle size. The answer is A. Okay. Next is 19 question number. Which of the following statement is not true for the biosensor? The answer biosensor consists of a vessel or series of vessels used to perform a desired conversion by enzymic means. The answer is D. Okay. Next is 20 question number. Which of the following feature is not possessed by biosensor? The answer is C. Active site is mainly constituted by non-polar amino acid for catalysis to take place. The answer is C. Next is 21 question number. Immobilized enzyme are more preferred over free enzyme in producing biosensor. The answer is true. Explanation करते हैं. Immobilized enzyme are more preferred over free enzyme in producing biosensor because of the following reasons. First, immobilized enzyme may be reused, ensuring the same catalytic activity for a series of analysis. B is immobilized enzyme are intrinsically established during the immobilization process. Then 3 is even if inactivation of enzyme occurs, it is steady or predictable. Then 4th is immobilized enzyme system are reusable up to 10,000 times over a period of several months. Next is 22 question number. Which of the following is the physicochemical component? The answer is C. Transducer. Explanation is transducer is referred to as the physicochemical component. Enzyme or antibody are biological components. Answer is C. Next is 23 question number. An example of biosensor urea electrode makes use of which of the following electrode? The answer is D. Ammonium electrode. Explanation. An example of biosensor urea electrode make use of the ammonium electrode. Urease membrane is also present. Answer is D. Next is 24 question number. Biosensor measure glucose concentration between which of the following range? The answer is D. 10 to 7 m. Okay. Explanation देखते हैं. Biosensor measure glucose concentration between 10 to 10 m. Okay. 10 के power 1 to 10 के power 7 m. Next is 25 question number. Which of these biosensor use the principle of heat release or absorbed by a reaction? The answer is D. Calorimetric biosensor. Explanation is calorimetric biosensor use the principle of heat released or absorbed by a reaction. The heat is measured to determine the concentration. The answer is D. Next is 26 question number. Nanoscopic optical biosensor have fast response time but the sensitivity is reduced. The answer is B. False. Explanation है nanoscopic optical biosensor have a fast response. They also have excellent biochemical sensitivity. The answer is B. False. Next is 27 question number. Given below is the diagram biosensor. Identify the unmarked component. जो unmarked component है इसमें क्या होगा? The answer is C. Transducer. Okay. Biosensor, then transducer, then amplifier, then processing, then display. Okay. Transducer explanation. The biological signal must be converted into electrical signal. Transducer are used for this purpose. Next is 28 question number. For constructing the glucose sensor, which of the following is used as a gel? Answer is D. Polyacrylamide. Explanation is for constructing the sensor, polyacrylamide is used as the gel. It is used to entrap the glucose oxidase. Answer is D. Next is 29 question number. Which of the following biosensors are used produced during redox reaction? The answer is A. Amperometric biosensor. Explanation is Amperometric biosensor uses movement of electrons. क्या movement करता है electrons produced during redox reaction. Redox reaction involve both reduction and oxidation. आज हम देखेंगे बायोटेक्नोलॉजी का कुछ टॉपिक्स का एमसीक्यू, जैसे कि फर्स्ट यूनिट का, जैसे कि प्रोटीन इंजीनियरिंग, यूज ऑफ माइक्रोब्स इन इंडस्ट्री एंड जेनेटिक इंजीनियरिंग, इसका एमसीक्यू देखेंगे। ओके, चलिए स्टार्ट करते हैं फर्स्ट क्वेश्चन नंबर से। फर्स्ट क्वेश्चन नंबर इज, ये पार्ट 2 है। ओके, फर्स्ट क्वेश्चन नंबर, which of the following technology offers new possibilities for the modification of natural proteins for a specific industrial, clinical or agricultural purpose? The answer is D. Protein engineering. Okay, इसका कुछ explanation देख लेते हैं। Protein engineering is a multidisciplinary technology for the design, क्या design and construction of protein. It offers new possibilities for modification of natural protein for a specific industrial, clinical or agricultural purpose. Okay, next is second question number, which is not the application of protein engineering. कौन से application इसमें protein engineering का नहीं है? Multiplication of natural protein. The answer is D. Okay, इसका कुछ explanation, multiplication of natural protein is a not a application of protein engineering, नहीं है protein engineering का application. Okay, next is third question number, random multigenesis followed by selection at the level of whole organism is a quick process. The answer is B. False. Okay, explanation देखते हैं, random mutagenesis followed by selection at the level of whole organism is a slow or laborious process. In a many way, it is the most successful protein engineering. Answer is B. Next is fourth question number, which of the following step is performed just after the protein form a natural organism is isolated? The answer is C. Preparative biochemistry. The answer is C. इसके कुछ explanation है, देख लेते हैं। Explanation is preparative biochemistry is the step that is performed just after the protein from natural organism is isolated. It is the biochemically characterized with a ligand that is as an enzyme, substrate, receptor, hormones or antibody antigen interaction. The answer is C. Next is fifth question number, in the protein engineering cycle, 3D structure of the protein ligands complex is determined by which of the following techniques? The answer is D. High resolution X-ray analysis. The answer is D. Explanation देखते हैं, in the protein engineering cycle, the three-dimensional structure of the protein ligands complex defined by the high resolution X-ray analysis or two-dimensional nuclear magnetic resonance. The short form is NMR. Okay. Next is sixth question number, which step is performed after the protein assay and characterization in the protein engineering cycle? The answer is A. Protein ligand complex analysis. The answer is A. Protein ligand complex analysis. इसका कुछ explanation देख लेते हैं। Protein ligands complex analysis is performed after the protein assay and characterization in the protein engineering cycle. In this step, the interaction of the target protein with its ligand is analyzed. The answer is A. Next is seventh question number, which of the following is not a step of the protein engineering cycle? Answer is D. Mass spectrometry. Explanation is mass spectrometry is not a step of protein engineering cycle. Gene cloning, protein assay and characterization and 2D NMR are some of the steps required in protein engineering. Okay. Next is 8 question number. Which of the following amylase is not a produced by organism? The answer is B. Hepatic amylase. The answer is B. Next is explanation is liver does not produce amylase and therefore there is no such hepatic amylase. However, saliva amylase, pancreatic amylase and gastric amylase are produced by the various organisms for the breakdown of a complex carbohydrate into a similar. And question number, the enzyme beta galactosidase is also known as both A and B. A is lactose, B is EC3.2 डियर फ्रेंड्स, तो आज हम देखेंगे यूनिट 2 बायोटेक्नोलॉजी का। ओके, जो एमसीक्यू ऑनलाइन एग्जाम जो हो रहे हैं उसमें ज़्यादा इंपॉर्टेंट है, तो आप स्टार्टिंग से लेकर के अंत तक देखिएगा, और जो भी प्रीवियस यूनिट है ना वो आपको डिस्क्रिप्शन लिंक में मिल जाएंगे। ओके, फर्स्ट क्वेश्चन नंबर देखते हैं। विच ऑफ द फ्लोइंग स्टेटमेंट इज ट्रू? कौन से इसमें है ट्रू स्टेटमेंट है? तो सारे ट्रू हैं। डी ऑल। ए इज अ वेक्टर should have a origin of replication. B is a vector should have selectable marker, and C is a vector should have unique restriction set. The answer is D. कुछ explanation है. A cloning vector is a small piece of DNA that can be stably maintained in an organism and into which a foreign DNA fragment can be inserted for cloning purpose. The answer is D. Next is second question number. Expression vector differ from a cloning vector in having. The answer is D. Control element. Element. The answer is D. Okay. Next is three question number. Combining the DNAs of two species into a single DNA molecules is called recombinant DNA techniques. The answer is B. Recombinant DNA technique explanation is comprises altering genetic material outside an organism to obtain enhanced and desired characteristics in living organism or as their products. The answer is B. Next is four question number. Which of the following does not act as a restriction enzyme? The answer is D. Polydeoxyribonucleotide synthesis. The answer is D. इसका कुछ explanation है. Polydeoxyribonucleotide synthesis is a DNA ligase. क्या है? DNA ligase है. Next is 5 question number. Which of the following enzyme is responsible for making a DNA copy or RNA? The answer is A. Reverse transcriptase. The answer is A. Reverse transcriptase. Explanation is reverse transcriptase generate complementary DNA and from RNA templates. The answer is A. Okay. Next is sixth question number. Which of the following can be termed as a restriction modification system? The answer is A. Restriction endonuclease plus methylase. The answer is A. Next, the explanation is restriction endonuclease plus methylase is termed as a restriction modification system. The answer is A. Okay. Next is 7 question number. Generally, a plasmid vector contains how many elements? The answer is C. 3 elements. Okay. Next is explanation. Cloning site, drug resistant, gene and replication origin are the 3 elements in the plasmid vector. Okay. Next is 8 question number. To make the recombinant plasmid permeable to DNA molecules, which of the chemical is added? कौन सा chemical इसमें added होगा? The answer is A. CaCl2. Okay. Explanation is the cell are treated with CaCl2 to make them permeable to DNA molecules through a process called transformation. The answer is A. Okay. Next is 9 question number. E core 1 is a. The answer is B. Restriction endonuclease. E core 1 is a restriction enzyme that cleaves vector at the cloning site. The answer is B. Next is 10 question number. In which stage of genetic engineering a probe is used? The answer is D. Screening. क्या screening is किया जाता है. The explanation is in hybridization, which of is one of the method of screening a probe used. Okay. Next is 11 question number. Which of the following enzyme is required for end to end joining of DNA? The answer is A. DNA ligase. Okay. Explanation is DNA ligase ligate both blunt and sticky end of the DNA. Okay. Next is 12 question number. Repressor molecules bind to operator. The answer is A. Operator. Explanation is An operator lies partially within the promoter. Repressure bind to the DNA at operator. The expression of gene. Okay, answer is A. Next is 13 question number. Which of the following is required for the preparing the phosphodiester backbone of a DNA during molecules cloning? The answer is D. DNA ligase. Explanation is DNA ligase is required for the preparing the phosphodiester backbone of DNA during molecules cloning. The answer is D. Next is 14 question number. A single strand of nucleic acid target tagged with a radioactive molecules is called. The answer is D. Probe. The explanation is probe are single stranded radio-labeled molecules of nucleic acid with a non-sequence. The answer is D. Next is 15 question number. For transformation of microparticles coated with the DNA to be bombarded with gene gun are made up of. The answer is C. Gold or tungsten. Explanation is the metal particle commonly used for the gene gun bombardment include gold, tungsten, palladium and rhodium, platinum and iridium. The answer is C. Okay. Next is 16 question number. The usual source of restriction endonuclease used in gene cloning is bacteria. The answer is B. Bacteria. Explanation is restriction endonuclease represent a class of nucleus originally extracted from the bacterium E. coli. The answer is B. Next is 17 question number. In genetic engineering, the antibiotic are used. The answer is A. As selectable markers. Explanation is antibiotics medium are used to mark or select transformant recombinant bacteria. So we can say antibiotics used as a selectable markers with a marker gene. The answer is A. Next is 18 question number. Which of the following is used to select gene of interest from the genomic library? The answer is D. DNA probes. Explanation is DNA probes are stretches of single-stranded DNA used to detect the presence of complementary nucleic acid sequence, target sequence by the hybridization. Okay, the answer is D. Next is 19 question number. The enzyme which are absolutely necessary recombinant DNA technology are. The answer is C. Restriction of endonuclease are and ligase. The answer is C. Okay. Next is 20 question number. Which of the following is a plasmid? कौन सा इसमें है plasmid? The answer is A. PBr322. Its explanation is PBr322 is a plasmid and was one of the first widely used E. coli cloning vector. Created in 1977 in the laboratory of Herbert Boyer at the University of California and San Francisco. The answer is A. Okay. Next is 21 question number. The TI plasmid is used for introduction gene into. The answer is B. Plant cell. Explanation is in plant genetic engineering the TI plasmid can be used carry foreign gene into the plant cells. The answer is B. Next is 22 question number. Which technique is used to separate protein fragments based on size? The answer is B. Southern blot. The answer is C. Aggregate gel electrophoresis. B is not right. C is right. Aggregate gel electrophoresis. Explanation: electrophoresis is laboratory techniques used to separate DNA, RNA or proteins molecules based on their size and electrical charge. Okay, answer is B. Next is 23 question number. Restriction endonuclease A cut the DNA molecules at specific sites. Explanation is restriction endonuclease cut the DNA molecules at a specific site. It is also called molecule season. The answer is A. Okay. Next is 24 question number. Polymerase chain reaction technology PCR it is used for the DNA amplification is the answer is true. Okay, true. Next is 25 question number. The bond obtained by gel electrophoresis is during separation and sustained. 1798 the first smallpox vaccine was developed. Answer is B. Okay. Next is 22 question number. The process of weakening and pathogen is called. The answer is B. Attenuation. The answer is B. Okay. Next is 23 question number. Which of the following statements are true regarding polio vaccines? The answer is D. All of these. A is Salk and Sabin are polio vaccines. B is Selvin is lab attenuated polio vaccines, and C is Salk is an inactivated polio vaccines. The answer is D. Next is explanation is in 1955 Salk developed in inactivated polio vaccines, poliovirus vaccines. Thus began widespread immunization. This was followed in 1960 by a lip attenuated oral vaccine developed by Sabin. Sabin से developed किया गया था oral vaccine. Okay, the answer is C. Sorry, D. Okay. Next is 24 question number. What is the ideal temperature of store of vaccines? कौन सा ideal temperature होता है store करने के लिए vaccines को किस डिग्री सेल्सियस में store किया जाता है? तो 2 से लेकर के 8 डिग्री सेल्सियस में आप स्टोर कर सकते हैं वैक्सीन को. Explanation: the vast majority of vaccine should be stored in between 2 to 8 degrees Celsius in a refrigerator with a preferred average of 5 degrees Celsius. Through some should remain frozen in a range between -15 to -50 degrees Celsius. Okay. Next is 25 question number. Who is the responsible for the cold chain management? The answer is E. All of this. A is the owner of the business, B is the nursing of a staff, and C is the medical staff, and D is the administrative staff. Okay. Next, the answer is E. All of this. Everyone has the role play. सभी को कोई रोल प्ले करता है इसमें. इसमें आपको g-patch रिवीजन क्लास को सब्सक्राइब करना है और शेयर करना है, लाइक करना है, कमेंट करना है. ओके, तब तक के लिए बाय. नेक्स्ट वीडियो में मिलते हैं. बात करेंगे यूनिट 3 बायोटेक्नोलॉजी से. ओके, 6M के लिए. और जो इम्पोर्टेंट है वो इसलिए क्योंकि जो ऑनलाइन एग्जाम जो आपके हो रहे हैं उसमें ज्यादा इम्पोर्टेंट है, तो स्टार्टिंग से लेके अंत तक देखिएगा. और जो यूनिट वाइज है वो आपको डिस्क्रिप्शन लिंक में मिल जाएंगे, आप वहां से भी देख सकते हैं. ओके, चलिए फर्स्ट क्वेश्चन नंबर देखते हैं बिना देर के हुए. फर्स्ट इज humoral immunity is mediated by, the answer is A, beta cell, B cell. Okay, B cell is also known as B lymphocytes, or type of a white blood cell of a lymphocyte subtype. They function in the humoral immunity components of an adaptive immune system. The answer is A. Okay, next is 2 question number. Humoral immunity is also called as, the answer is B, antibody mediated immunity. The answer is B. Okay, next is 3rd question number. B cells produce antibodies in response to the instruction received from, the answer is D, helper T cells. Explanation is B cell produce antibodies in response to the instruction received from helper T cells. Along with this, helper T cells also attract macrophages to the site of infection. The answer is D. Next is 4 question number. Which of the following statement is incorrect regarding plasma cells? The answer is D. Plasma cells has surface receptor. Okay, the answer is D. Next is 5 question number. Generally in antibodies produced against the pathogen is polyclonal. The answer is C, polyclonal. Explanation is polyclonal antibodies are antibodies that are secreted by different B cells ligands within the body. They are the collection of immunoglobulins molecules that react against a specific antigens. The answer is C. Okay, next is 6th question number. Maps are The answer is B. Specific toward an epitope. Epitope. The answer is B. Explanation is monoclonal antibodies are cells derived by cell division from a single ancestral cells. Monoclonal antibody is specific to a single epitope. The answer is B. Next is 7 question number. MAB are used in, the answer is D, all. The A is screening of recombinant, B is diagnostic kit, and C is the treatment of many cancers. The answer is D, all of this. Next is 8 question number. MABs are produced by, the answer is D, all of these. A is in vivo method, B is suspended in cell culture in fermenter, then C is immobilized cell reactor. The answer is D. Okay. Next is 9 question number. What is the name of MHC in human? The answer is A. HLA. Explanation is the gene of MHC major histocompatibility complex are located in the short arm of chromosome in 6 in humans. In human, it is also known as human leukocyte antigen. The short form is HLA. Okay. Next is 10 question number. Which of the following polypeptide is important for the expression of MHC1 on the cell membranes? The answer is B. Beta 2 microglobulins. The answer is B. Explanation is beta microglobulins is encoded by chromosome 15. It is always attached to alpha 3 subunit of MHC1 molecules through non-covalent interaction. The answer is B. Next is 11 question number. Name the cell which receive antigen presented by the MHC molecules. The answer is C. T cells. Okay. Explanation is MHC molecules processed antigens to T cells as T cell receptor does not recognize free antigen. It will recognize only those antigen which are bound to MHC molecules. The answer is C. Okay. Next is 12 question number. Name the class of MHC which is recognized by CD4-TH. The answer is B. MHC2. Okay. MHC2 explanation is CD4 is a monomeric single chain which recognize MHC2 molecules and is present on T helper cell. It consists of a core extracellular domain and has 3 critical serine residues on a cytoplasmic tail. The answer is B. Next is 13 question number. Which of the following statement is incorrect about the super antigens? The answer is B. Endogenous by nature. Explanation is super antigen are viral or bacterial proteins which have the unique binding ability of T cell receptor and MHC2 molecules. That is, it binds simultaneously to both an active large members of T cells. It can be both either endogenous or exogenous in nature. The answer is B. Okay. Next is 14 question number. What is the life of Selector cell of the immune system? The answer is A. A few days. Explanation is the lifespan of effector cell of the immune system is few days only. Our body releases 2 types of cell effector. Memory cells of which effector cells are short-lived and are responsible for the primary response of our body. The answer is A. Next is 15 question number. Allergy to penicillin is an example of, the answer is A, type 1 hypersensitivity. The answer is A. Explanation is type 1 hypersensitivity or immediate hypersensitivity is an allergic reaction provoked by re-exposure to a specific type of antigen referred to as an allergen. The answer is A. Next is 16 question number. Type IB hypersensitivity is also called as, the answer is B, delayed hypersensitivity. The answer is B. Explanation is टाइप 1 hypersensitivity ये होता है immediate hypersensitivity. टाइप 2 जो होता है ये cytolytic reaction होता है. Then टाइप 3 जो होता है immunocomplex hypersensitivity होता है. Okay. Next is 17 question number. The most common classes of antibody involved in type 2 hypersensitivity is, the answer is D, both A and B. A is IgG and B is IgM. Okay. Explanation is the antibodies involved in a type 2 reaction are both IgM and IgG. Okay. Next is 18 question number. T helper cell mediated hypersensitivity is, the answer is D, type 4 hypersensitivity. Okay. The answer is D, type 4 hypersensitivity. Explanation is often called delayed type of hypersensitivity as the reaction takes several days to develop. This response involves the interaction of T cells, monocytes and macrophages. The answer is D. Next is 19 question number. Type 3 hypersensitivity is triggered by, the answer is C, deposition of antigen antibody complex. The answer is C. Explanation is in type 3 hypersensitivity reaction an abnormal immune response is mediated by the formation of antigen antibody aggregate called immune complex. The answer is C. Next is 20 question number. Will and flare reaction is characteristic reaction associated with identification of, the answer is A, type 1 hypersensitivity. ये क्या होता है? Called a will and flare reaction is include swelling मतलब फूलना produced by the release of serum into a tissue well and redness of the skin redness आ जाता है skin पे and resulting from the dilation of blood vessels flare उसको क्या बोलते हैं? Flare. Okay, next is 21 question number. The first vaccine was developed by, first vaccine जो था वो develop कौन किया था? Edward Jenner. Explanation is Edward Jenner is considered the founder of vaccinology in the West in 1796 after he inoculated a 13-year-old boy with vaccinia virus cowpox and demonstrated immunity to a smallpox. 1798 the first smallpox vaccine was developed. Answer is B. Okay. Next is 22 question number. The process of weakening and pathogen is called, the answer is B, attenuation. The answer is B. Okay. Next is 23 question number. Which of the following statements are true regarding polio vaccines? The answer is D, all of these. A is Salk and Sabin are polio vaccines. B is Selvin is lab attenuated polio vaccines and C is Salk is an inactivated polio vaccines. The answer is D. Next is explanation is in 1955 Salk developed in inactivated polio vaccines, poliovirus vaccines. Thus began widespread immunization. This was followed in 1960 by a lip attenuated oral vaccine developed by Sabin से developed किया गया था oral vaccine. Okay, the answer is C, D. Okay, next is 24 question number. What is the ideal temperature of store of vaccines? कौन सा ideal temperature होता है store करने के लिए vaccines को? किस डिग्री सेल्सियस में store किया जाता है? तो 2 से लेकर के 8 डिग्री सेल्सियस में आप store कर सकते हैं vaccines को. Explanation: the vast majority of vaccine should be stored in between 2 to 8 degrees Celsius in a refrigerator with a preferred average of 5 degrees Celsius. Through some should remain frozen in a range between -15 to -50 degrees Celsius. Okay. Next is 25 question number. Who is the responsible for the cold chain management? The answer is E, all of these. A is the owner of the business, B is the nursing of a staff, and C is the medical staff, and D is the administrative staff. Okay. Next, the answer is E, all of these. Everyone has the role play. सभी को कोई रोल प्ले करता है इसमें। इसमें आपको जी-पैट रिवीजन क्लास को सब्सक्राइब करना है और शेयर करना है, लाइक करना है, कमेंट करना है। ओके, तब तक के लिए बाय, नेक्स्ट वीडियो में मिलते हैं। आज का जो एमसीक्यू है वो बायोटेक्नोलॉजी से है, जो कि यूनिट 4 है और इम्पोर्टेंट इसलिए क्योंकि जो ऑनलाइन एग्जाम हो रहे हैं उसमें ज्यादा इम्पोर्टेंट है। तो स्टार्टिंग से लेकर अंत तक देखिएगा और सारे यूनिट का आपको डिस्क्रिप्शन लिंक में मिल जाएंगे, आप वहां से भी देख सकते हैं। ओके, चलिए स्टार्ट करते हैं। फर्स्ट क्वेश्चन नंबर, वेस्टर्न ब्लॉटिंग इज द टेक्निक फॉर द डिटेक्शन ऑफ, the answer is C, a specific protein in a sample. The answer is C. Okay, next is second question number, labeled antibodies are used to detect, labeled antibodies are used to detect the presence of a particular protein molecule in western blotting. The answer is D. Okay, next is third question number, which of the following technique is used in DNA fingerprinting? DNA fingerprinting. The answer is B, Southern blotting. The answer is B. Okay, next is fourth question number, arrange the following in correct order. क्या करेक्ट ऑर्डर में इसको अरेंज करना है। इसका जो आंसर होगा वो बी होगा, 1 का बी, 2 का 1 का बी, 2 का D, then 3 का A and 4 का C. This is the correct order. Okay. The next is 5 question number, arrange the following in correct order. इसको भी correct order में करना है। इसका जो आंसर होगा वो B होगा, 1 का B, तो 1 का B ये लग जाएगा, B सेकंड का D, then सेकंड का D, 3 का A, then 3 का A and 4 का C. 4 का C. Okay, this is the correct answer. Okay, next is 6th question number, which of the following technique is suitable for the identifying mRNA molecules in a sample? The answer is C, Northern blotting. The answer is C. Next is 7th question number, which of the following technique does not involve electrophoresis for the separation of biomolecules? The answer is A, dot blotting. This is the dot blotting. Okay, the answer is A. Next is 8th question number, props is a short piece of labeled DNA or RNA which are complementary to the nucleic acid standard to be detected. The answer is C. Okay, next is 9th question number, which of the following technique most detecting the presence of gene product? The answer is D, Western blotting. The answer is D. Next is 10th question number, aminobenzyl benzoxymethyl filter paper is commonly used for the transfer in. The answer is C, Northern blotting. The answer is C. Next is 11th question number, vertical gene transfer is the process of gene being passed down from parent to offspring. The answer is A, true. The answer is A. Next is 12th question number, when bacteria take up genetic material from a surrounding environment, this is called transformation. This is called transformation. The answer is A. Okay, next is 13th question number, when one bacterium transfer a copy of its plasmid to another bacterium, this is called conjugation. The answer is C, conjugation. Okay, next is 14th question number, transformation, transduction and conjugation are all example of. The answer is A, horizontal gene transfer. The answer is A. Next is 15th question number, transposable elements are often found once in a gene and have a unique sequence for its own. The answer is false. Explanation is the transposable element have the ability to move from one location to the another within the genome and often make duplicate copies of them in this process. Answer is B. Okay. Next is 16 question number. Transpons were first discovered in, the answer is D. Mass. Explanation is the first transpons were discovered in Mass in 1948. For this discovery, Barbara McClintock was awarded a Nobel Prize in 1983. Okay, the answer is D. Next is 17 question number. The type of mutation that is the imposed by transpon is, the answer is C. Polar mutation. The answer is C. Okay. Next is 18 question number. Transpon are able to move by two mechanisms. Moved by two mechanisms. Explanation is transpon can be moved by conservative or replicative process. The answer is B. 2. The next is 19 question number. Silencing the transpon cannot be achieved by which of the following mechanism? Chromatin. Sorry. The answer is C. DNA phosphorylation. The answer is C. Okay. D is not. The answer is C. DNA phosphorylation. Next is 20 question number. In Griffin's experiment, which of the following strains of pneumococci was isolated from dead mice? The answer is C. Live smooth cells. The answer is C. Next is 21 question number. Which of the following things was identified as the transforming principle? The answer is A. DNA. The answer is A. Next is 22 question number. What should be the minimum weight of DNA that is the required for the successful transformation? The answer is C. 3 lakhs daltons. The answer is 3 lakh daltons. Okay. Explanation is molecular weight of DNA in the range of 3 lakh to 8 million daltons have been shown to result in successfully transformation. The answer is C. Next is 23 question number. In which phase of growth does the recipient cell take up the donor DNA? The answer is C. Late logarithmic phase. The answer is C. Next is 24 question number. Mark the incorrect statement about mutation. Incorrect statement इसमें कौन से हैं? The answer is A. Mutation is predefined. The answer is A. Next is 25 question number. Addition or deletion of base cause which kind of mutation? The answer is B. Frame-shift mutation. The answer is B. Next is explanation. Frame-shift mutation is a point of mutation involve the addition or deletion of base while substitution of base cause transition or transversion. The answer is B. Okay. नेक्स्ट आपको क्या करना है? नेक्स्ट जी-पैट रिव्यूइंग क्लासेस को सब्सक्राइब करें और शेयर करें और लाइक करें, कमेंट करें. तब तक के लिए बाय. नेक्स्ट हम बात करेंगे यूनिट 5 की बायोटेक्नोलॉजी से, जो कि इम्पोर्टेंट है. आपको जो ऑनलाइन एग्जाम हो रहे हैं उसमें ज्यादा इम्पोर्टेंट है. तो आप स्टार्टिंग से लेकर अंत तक देखिएगा और सारा लिंक आपको डिस्क्रिप्शन में मिल जाएंगे. आप वहां से भी देख सकते हैं सारे यूनिट्स का. ओके, चलिए स्टार्ट करते हैं. फर्स्ट क्वेश्चन नंबर इज व्हाट इज द बेसिक फंक्शन ऑफ द फर्मेंटर? द आंसर इज सी. टू प्रोवाइड ऑप्टिमम growth condition to organism and obtain the desired product. The answer is C. Okay. Next is second question number. While constructing the fermenter, which of the following is not required? The answer is A. High speed aggregation and aeration system. The answer is A. Next is third question number. Which of the following material is preferable for constructing of a small scale fermenter? The answer is B. Glass. Explanation is the glass is most preferred material for the construction of fermenter. It is transparent so that it is easy to visual the interior of the glass vessel. Moreover, the glass is non-toxic and corrosion proof. The answer is B. Okay. Next is fourth question number. Which of the following is not the technique of preservation? The answer is D. The storing water. The answer is D. Next is five question number. The preservation of by liquid nitrogen is called cryopreservation. The answer is A. Cryopreservation. The answer is A. Next is sixth question number. Which of the following is not a cryoprotective agent? The answer is D. Paraffin wax. क्या है? नहीं है cryoprotective agent. तो कौन सा नहीं है? Paraffin wax. Explanation is the cryoprotective agent like DMSO, 10%, glycerol, glycol are the agents that are added to microbial cell culture to protect the cell at a lower temperature in liquid nitrogen at minus 9196 degrees Celsius. Okay. Next is seven question number. Which of the following is used dried culture preservation? The answer is C. Both A and B. A is sand, B is soil. Okay. Next is eighth question number. During the preservation of microbial cell culture, the answer is B. Metabolism continues. Explanation is the preservation of microbial cell culture does not alter the metabolism of microbes. The answer is B. Next is ninth question number. Which of the following method of preservation is more suitable and used widely? The answer is B. Lyophilization. The answer is B. Next is 10 question number. Which of the following is not the replacement in serum free media? The answer is C. Antibody. Explanation is the important placement in serum free media are insulin, albumin, transferrin, ethylenamine, selenium, and B-mercaptoethanol. The answer is C. Next is 11 question number. Hybridoma technology is used in the production of. The answer is C. Monoclonal antibodies. The answer is C. Next is 12 question number. Which of the following provides a protective effect to animal cells? The answer is B. Pluronic F28. Okay. Next is 13 question number. Which of the following is downstream processing? The answer is C. Product recovery. Explanation is the downstreaming processing includes purification and recovery of product obtained from fermenter. The answer is C. Next is 14 question number. Which of the following is not a type of sterilization? The answer is D. Submerged. The answer is D. Submerged is a type of fermentation where microorganisms are grown in liquid medium. The answer is D. Next is 15 instrument works on the principle of batch sterilization. The answer is B. Autoclave. The answer is B. Next is 16 question number. The highest feasible temperature for batch sterilization is. The answer is B. 121 degrees Celsius. The answer is B. Next is 17 question number. The holding time at 135 degrees Celsius will be. The answer is B. 51.9 seconds. 2.44 minutes was the holding time at 130 degrees Celsius. 18.9 seconds was holding time at 140 degrees Celsius and 2.7 second was holding time at 150 degrees Celsius. Okay. Next is 18 question number. Which of the following is a type of continuous sterilizer? The answer is A. Steam injector. The answer is A. ये होता क्या है? Explanation is there are two types of continuous sterilizer: indirect heat exchanger and direct heat exchanger or steam injector. Okay. Next is 19 question number. Which of the following sensor is used to measure the change in flow rate? The answer is B. Weight. Next is 20 question number. Which of the following device is not used to pressure measurement? The answer is D. Dynamometer. Dynamometer is useful in aggregation measurement and control. Okay. Next is 21 question number. Antiform probe is a type of offline sensor. The answer is B. False. Explanation is the antiform probe is a type of inline sensor. They are an integral part of the fermenter. It is used to check the formation in the fermenter and control it. Okay. Next is 22 question number. Penicillium chrysogenum produce penicillin. The answer is A. True. Explanation is Penicillium chrysogenum is a species of fungus. Common in temperate and subtropical region, it is the source of several beta-lactam in antibiotics, most significant penicillins. The answer is A. Next is 23 question number. pH required for the production of penicillin will be, the answer is B. 6.5. Explanation is the pH should be between 6.4 and 6.8 during active production phase of penicillin. Okay. Next is 24 question number. Who first isolated citric acid? The answer is B. Carl William. कौन से आइसोलेटेड किया था सिट्रिक एसिड? फर्स्ट में कार्ल विलियम ने किए थे. एक्सप्लेनेशन इज कार्ल विलियम इन 1784 में फर्स्ट आइसोलेटेड किया था सिट्रिक एसिड को एंड फ्रॉम लेमन जूस से. Citric acid is a tribasic acid. Okay. Next is 25 question number. Who observed citric acid as a byproduct of calcium oxalate produced by Penicillium glaucum? The answer is C. Weimar. Okay. Answer is C. Weimar in 1893 observed citric acid as a byproduct of calcium oxalate produced by Penicillium glaucum. The answer is C. Next is 26 question number. Which of the following species is used for the vitamin B12 production by fermentation process? The answer is D. All of these. A is Streptomyces gracis and B is Streptomyces ovalaceus and C is Bacillus megaterium. The answer is D. All of these. Next is 27 question number. Which of the following is the most common method for citric acid production? The answer is B. Submerged fermentation. The answer is B. Next is 28 question number. Which of the following raw material are important for the production of glutamic acid? Glutamic acid. The answer is D. Biotin. Explanation is for glutamic acid is medium generally consists of the carbohydrate, peptone, inorganic salts and biotin. The answer is D. Next is 29 question number. Gracefulin is a secondary metabolite produced by Penicillium gracefulin. Yes, gracefulin is a secondary metabolite produced by Penicillium gracefulin and isolated in 1939. Okay. Next is 30 question number. Which of the following is the most common method for gracefulin production? The answer is A. Solid state fermentation. The answer is A. Okay. The next is g-patrevision classes को सब्सक्राइब जरूर करें एंड शेयर करें, लाइक करें, कमेंट करें. ओके. तब तक के लिए बाय. नेक्स्ट वीडियो मिलते हैं.